Welcome back. Here in section 2, we will consider the concept of gene design, which is fundamental to synthetic biology. Redesigning genes does not mean creating artificial life. However, because our genes are mainly composed of DNA, chemical synthesis of DNA, or even the creation of a synthetic genome or life with a synthetic genome is theoretically possible. For example, as in this case of the creation of the first cell with a synthetic genome, if you click the video of a featured news clip, they called it a synthetic cell. However, this is not actually a synthetic cell. It is a synthetic genome. We shall discuss the creation of Synthia, a bacterium with a synthetic genome, in section 3, and here we start from the concept of what is in a gene to explain a bit more about designing genes. What's in a gene? We need to know the structure, function, and regulation of a gene to make it work under our control. Here, we draw the line of DNA. A gene should consist of a gene promoter, a translational start site, the gene coding region, and a terminator. Can you determine the exact locations of these components? If the transcription start site is there, the order 1, 2, 3, 4 is correct. Beginning with the gene promoter, a translation start, after the transcription start, the coding region, and a terminator. Of course, this is a typical case, and in some special cases, the promoter could be downstream of a gene, and for eukaryotic cells, most genes also have introns inside the coding region. We usually use a plasmid as a vector to carry the designed gene or DNA. A plasmid is circular DNA outside the genomic DNA or chromosome. PBR322 or PUC PUC vector here that has 3 major components: a region of replication, multiple cloning sites for insertion of the gene, and an antibiotic resistant gene. The PUC vectors have beta-galactosidase gene or BGAL as a marker gene behind the multiple cloning sites. The inserted DNA can interrupt the BGAL gene reading frame, and thus the produced enzyme malfunctions and produces no color with white colony even in the presence of a substrate called a scale, which normally gives blue colors. In a gene cassette, we can put different gene segments together with compatible restriction enzymes, exemplified here as A, B, C, D. We can join 3 segments of DNA or join a gene promoter to a coding region to detect pollution or degrading toxic chemicals. Of course, we can also make useful products. You may check the iGEM student handbook for details for this link. iGEM, which stands for International Genetically Engineered Machine, is a competition held every year in Boston. Here is an example of a promoter with promoter A1 in device A to produce a product of A2 that could induce the production of B2 in device B by inducing promoter B1. Such amplification effects are a common design in synthetic biology that can enhance the production of the products you want. A profound amplification effect should result from these two levels of control of gene expression. Another example here is a report on redesigning a gene to turn on promoter genes known as green fluorescent protein, GFP. They also add an enzyme gene called LAC alpha in front of the GFP. LAC alpha is an enzyme called galactosidase that can turn the substrate blue. Here they test the gene promoter of J23119 as the strongest promoter when transfected into Chinese hamster ovary cells or CHO cells, and it could be visualized under microscopes with both GFP and blue colors. Like this Taiwan team, they designed a gene cassette to sense a metal ion called lead, with a lead binding protein which is engineered to act on a gene promoter to produce green fluorescent protein, GFP. Similar idea goes to detect arsenic, copper, and other toxins in T samples. In 2010, the Cambridge University iGEM team expressed a very strong fluorescent signal in bacterial culture. Look at this flash of bacterial culture expressing the fluorescent protein and emitting a strong, bright light. The engineered E. coli is called E. coli. Sounds interesting, right? A good example here is the use of DNA to detect the chemical pollutant arsenic. Many different teams worked on this idea, from Edinburgh University in 2006 to a Cornell team in 2012. They even developed a test kit with arsenic receptors to sense the presence and amount of arsenic. In the gene cassettes for the arsenic sensor system, lactose normally induces the production of activator A1 to induce a gene encoding urease, which is an enzyme to produce ammonia. In the presence of arsenic, the reported genes include a repressor gene to repress the production of urease, and with higher concentrations of arsenic, LAC will be produced to create lactic acid. By putting all these different redesigned genes together in cells and experimenting with the ammonia and lactate being produced, we can use color agents to reflect acidity or pH values. Recent teams even tested the pH ranges or pH changes over time with different concentrations of arsenic relevant to WHO guideline levels of arsenate. A good dose-response relationship was detected at 600 and 1000 minutes, and this arsenic detection kit is now available commercially for field testing of arsenic contamination in water or soil samples. Let's take a break now. In the next section, we shall look at the details of the synthetic life form called Synthia and other genetically engineered machines.